Good morning, my dear friends. This is Sudhindra, working as assistant professor in the College of Dairy Science, Amreli, district Gujarat. Today, I am here to discuss about total viable count of milk. This total viable count of milk is very much important in the dairy industry, as we all know that microorganisms are responsible for spoilage of milk and milk products. Using this method, we can check the number of live microorganisms that are present in milk. This technique is otherwise called as standard plate count test, total plate count test, aerobic plate count test, or total bacterial count. This test employs a serial dilution technique. The serial dilution technique can be explained using this small picture. In this, we take 1 ml of milk into 9 ml of phosphate buffer or a buffer solution and thoroughly mix it. Again, we take 1 ml of milk of this dilution into second test tube containing 9 ml buffer solution, and this will become second dilution. Again, from here we add third into the third test tube containing 9 ml of buffer. So we will be adding successively into buffer. So we will be gradually decreasing the, means we will be diluting 10 times every time we add into the next buffer solution. After preparing the dilution, we mix it with the sterile nutrient medium in a petri plate and incubate it at suitable temperature. Here we will be incubating at 35 to 37 degrees centigrade for 24 to 48 hours. It is said that each bacterial colony that is developed on petri plate will be grown from a single bacteria, and this test will give best results when the bacterial population is very low. Especially when we do this experiment for pasteurized milk, we get best results, as the pasteurized milk should not contain not more than 30,000 colony forming units per ml as per the standards. Here it is a pictorial diagram of the dilution. As earlier said, first dilution, we prepare first dilution, then second dilution, then third dilution, then fourth dilution, then fifth dilution. This first dilution will be transferred to 1 ml of first dilution will be transferred to petri plate, 1 ml of dilution will be transferred to petri plate. Similarly, we will be transferring 1 ml of the dilution into petri plates. This is one technique. This is another technique wherein we add, while adding into second dilution, we take 1 ml directly into petri plate and 1 ml into second dilution. Both techniques are good, but here we will be reducing our time of experiment in this process. After adding the dilution, we add the nutrient media or nutrients that are to be provided for the bacteria in the form of agar and allow it to solidify. After solidification, we transfer these petri plates into the incubator at 35 to 37 degrees centigrade for 24 to 48 hours. After 24 to 48 hours, we will see different colonies on the petri plate. There will be different morphological characteristics of the colonies that are present. Basing on those morphological characteristics, we can guess what kind of bacteria or molds or fungi are present in the milk. Here, this picture will be best depicting how the dilution will be helpful in counting the number of bacteria. In the second dilution, if we see, we cannot analyze or estimate or there won't be any number of microorganisms in this. As we dilute to fourth dilution, here we can see the colonies, but it is very difficult for us to count. But as we go further into sixth dilution, here we can see individual specific number of microorganisms, and as said earlier, each colony will be forming from a single bacterium. So if we count these individual colonies, we can say this many number of bacteria are present in that specific dilution. When coming to advantages of standard plate count test, we can guess the number of live microorganisms that are present. It is possible for us to check differential counts, means we can see the difference of bacteria present based on the morphological characteristics. We can check, we can guess what kind of bacteria is present, and this method is helpful in grading the pasteurized milk based on the number of bacteria that are present, as well as to check the quality of the processing conditions during each step of milk processing of milk. And when we see the disadvantages of standard plate count test, it is just a rough estimate, because in this we only get the microorganisms that are able to grow at 35 to 37 degrees or a mesophilic count. We won't be able to get a count on psychrotropic organisms as well as thermophilic organisms. This is a very time consuming, laborious and cumbersome method which requires at least 24 hours. And this test requires huge number of reagents, huge quantities of reagents and chemicals, glassware at for performing in single time. And finally, most important thing is the person who is carrying out this test should be an expert. Otherwise, the results that we get will be not proper. Thank you. Hope I have made my point clearly to you.